Hello, everyone, and welcome to the November edition of MycoTalks. It's my pleasure to be here, and I'll co-chair this with Jay Coles. So we have two fantastic speakers today: Eric Dang from the Ragon Institute of Mass General, MIT, and Harvard, and he will talk about barrier tissue immunity to pathogenic fungi. And we have Minh-Hong Nguyen from University of Pittsburgh, who will be our second speaker. So the MycoTalks is a series that we started now several years back. The philosophy is diversity in career stage, gender, geography, field. We have 2 30 minutes MycoTalks. We keep this format. It's been successful so far. 30 minutes of in-depth discussion, and after that we have Q&A after the second presentation. So please put your questions in the Q&A sections, and we will read them and ask our speakers. The January MycoTalk will be by two presenters, being Zai and Bern Hube, and then we have the February one by George Dippy and Tamara Doering. I want to also advertise here another a series of talks, MycoClinics, which we also started 1 year ago. And there will be a December edition, December 11th. And for those of you who are interested in clinical mycology, please join that. So without further ado, I'll start with our presenting our first speaker. So that's Dr. Eric Dang. Eric performed his graduate work at the University of California, San Francisco, in the laboratory of Jason Cyster. There he studied oxysterols and cholesterol metabolism in regulating macrophage inflammatory responses. He received his Ph.D. in 2018, and soon after he joined the laboratory of Hiten Madhani at UCSF for his postdoctoral work, where he did some very seminal work using genetic approaches, where he studied the mechanisms of immune system manipulation by Cryptococcus neoformans. He had some very seminal findings in Th2 immunity in the lungs. It's a fantastic study. In 2022, Eric joined a tenure-track position at NIAID in the laboratory of host immunity and microbiome. And then in 2025, he moved at the Ragon Institute in Cambridge, Massachusetts, where he holds an academic appointment in the Department of Immunology at Harvard Medical School. His group focuses on understanding the crosstalk between fungal pathogens and commensals and the mammalian host. And with that, we are looking forward to Eric's presentation.
All righty. Is that the right screen share mode? There we go. I think that works.
Yes, looks like it.
Thanks so much for the introduction, Ilian. And thank you to the organizing committee for this opportunity to present. I wanted to show a sort of news story from the lab that's sort of focused a bit more on the intestine, when a lot of what we've been doing is in the lung. So I usually give an opening slide with a bit more of a fungal pitch, but I think for the global myco community, everyone's already drinking the Kool-Aid. But I just want to open up by making this point that I think we all appreciate that fungi are a present but ever-growing source of human morbidity and mortality. And beyond pure immunosuppression, I think there's an interesting concept that's emerging in the field, which is that fungal infections can result from maladaptive immune decision-making. And what I mean by that is that we've now sort of appreciated in immunology over the last 30 years that immune responses have to be exquisitely tailored to the appropriate pathogen that's the invading insult, where we turn on selective classes of responses that try to clear those selective agents. And so each of these immunological arms has a physiological role, So type 1 immunity is generally characterized by interferon gamma producing lymphocytes that try to ramp up cell autonomous killing mechanisms to control the growth of intracellular bacteria, viruses, fungi, as well as tumors. Type 2 immunity is characterized by the production of IL-13 and 4 from GATA3 positive lymphocytes, and this acts to protect against parasites and allergens through induction of the classic weep and sweep response that tries to expel these noxious agents. And then lastly, there's type 3 immunity, which is characterized by mostly IL-17 production, and this has been very clearly shown to provide protection against extracellular bacteria and fungi. Now, when I say that infection can be driven by maladaptive decision-making, what I mean is that it's now appreciated that beyond just purely immunosuppression, that turning on the inappropriate class of response can actively predispose to fungal colonization or invasive disease. And I think one of the best examples of this comes from my colleague at NIH, Mihalis Lianakis, where what he has beautifully shown is that in addition to IL-17 deficiency, overproduction of interferon gamma, particularly in the skin, actively promotes colonization of mucocutaneous candidiasis. There's also data that's been present in the field for many decades now, which shows that a number of environmental fungi have this predilection to turn on type 2 immunity in the lungs when they're inhaled. And this flavor of immune response is actively immunosuppressive to the ability of the host to clear the microorganism. And so one of the major questions that my lab is really interested in is trying to understand how the immune system makes decisions to turn on physiological versus maladaptive immune responses in the face of fungal exposure. We're particularly interested in this process of type 2 inflammation turned on by fungi. And one thing that we debate a lot in the lab is how much of this predilection to turn on type 2 immunity is a pathogen-driven response, meaning the host is being manipulated, versus this being a conserved evolutionary response. And I think that the idea that it's pathogen-driven would be kind of neat, particularly for things that we think about. But I view it that if you actually look more at the broad literature, this is a much more conserved functionality. And so it's very clear that environmental molds, so things like Alternaria, Penicillium, Aspergillus, when these are inhaled, they predispose to type 2 inflammation. And many asthmatic patients, up to 20 to 80%, depending on the studies, show that there's IgE sensitization against fungal allergens. There's also some really interesting emerging data on commensal, or at least gut fungi, that when these show dysbiosis, particularly in early life windows, that this can predispose to type 2 inflammation both locally, but also have distal effects that enhances airway inflammation. And that's actually a bit of what I'm going to be talking about today. And then lastly, there are these environmental yeasts that can drive type 2 immunity, I think, through these conserved initiation pathways, but then some of them have effector molecules that can hijack this response. But I think broadly, this is not a microorganism-directed pathway. This is a host-directed pathway. And so largely what my lab is interested in is sort of looking at the interface of mycology and immunology to try to understand the inputs and outputs of fungal-induced type 2 inflammation. And we approach this from 3 general questions. So one is trying to understand the repertoire of fungal cues that influence host immunity. 2 is trying to understand how type 2 inflammation is initiated in response to fungi. And then lastly, we're interested in understanding when type 2 responses are turned on, why is this maladaptive? And so what I want to talk about today is an ongoing project that sort of merges question 1 and 2. And our sort of lead into this was something that I just alluded to, which was some of these human epidemiologic studies that have linked fungal dysbiosis to asthma sensitization. Some of this comes from studies that have correlated asthma incidence with maternal or early life antibiotic use. And there's been hypotheses that that may be due to depletion of bacteria and enrichment of fungi. There have also been some nice studies done by Brett Finley's group in Vancouver, where they've looked at cohorts of kids that either developed asthma or not. And one thing that they found is that kids that develop asthma later in life showed early fungal dysbiosis in the guts around age 2 to 5. And then again, I just want to bring back to this point, which is that a lot of asthma patients do show sensitization to fungal allergens. And so from this sort of human backbone, one thing that's been interesting to think about is, can this be mimicked using animal models? And I think from there, it's fitting to get introduced by Ilian, because it was a study from his group and in parallel some work from David Underhill that initially sort of got at this. And so what they published was this really cool experiment where they put mice on fluconazole drinking water to try to deplete gut bacteria. and then come in with different house dust mite challenges to then drive a type 2 response in the lungs. And what they found was that giving mice fluconazole actually resulted in worsened allergic airway disease, which sort of might argue that fungi are actually protective in this context. But when they actually looked at the gut composition of fungi, what was clear is that you get depletion of fluconazole-sensitive strains like Candida, but you actually get expansion of fluconazole-resistant fungi, one example being here Aspergillus amstellodamii in green, but then really one that shows almost no colonization that then expands dramatically was the species called Willemia sebii, or also now known as Willemia mellicola. And then what was subsequently shown is that if you just monocolonize with Willemia and then give house dust mite, that gut colonization by Willemia is sufficient to enhance an airway eosinophilic response. And so our sort of starting framework then is trying to understand not these distal effects per se, but how are the fungi initially being sensed in the gut in order to initiate type 2 inflammation? And so some of this then comes to this paradigm now of epithelial alarmin production as the upstream driver of type 2 initiation. And so what's appreciated is that in response to allergen challenge, epithelial cells make a trio of innate type 2 cytokines, one of the classic ones being IL-33, also TSLP, and then the newer player is IL-25. Each of these three will signal to resident lymphocytes or myeloid cells in the tissue, and this serves to initiate type 2 responses. One thing that we were sort of thinking about on these alarmin side is, how do you get this crosstalk between the gut and the lung, particularly because these resident lymphocytes, like innate lymphoid cells, are thought to be non-recirculatory. And there was this paper from Ron Germain's group in 2018 where they had confirmed that at homeostasis. So if you stain for ILC2s here in red in an unchallenged mouse, they stay in the tissue. They don't exchange between parabionts. And you see they very rarely show up in these cyan villus lacteals, which are the draining lymphatics. But upon injection of IL-25, one of these epithelial alarmins, you see ILC2 expansion, and then they start showing up in these lacteals where they egress from the tissue. And then if you look by flow cytometry in the blood, normally they're almost non-existent in the blood because they're non-recirculatory, but after IL-25 challenge, they start showing up in the blood, and they also start showing up in distal organs like the lung. And so this idea of epithelial alarmin production might be a mechanism whereby you now drive systemic recirculation of lymphocytes that are normally tissue resident, and that can enhance distal airway responses. And that's actually been followed up by a very recent paper where they showed that if you colonize with Trichomonas murus in the gut, this drives recirculation of ILC2s, and that effect has an enhancement on house dust mite asthma in the lung. But again, for some of the questions we were thinking about then, if IL-25 can have this functionality, well, who are the cells actually making 25, and how does that work? And this kind of comes to some really interesting new epithelial biology that's emerged over the last decade, which has found that tough cells, this very rare and interesting cell type in the intestine, are the major source of IL-25 in vivo. So there were 3 papers that came out about 10 years ago that all found this. This is the data from Rich Loxley's paper where they made an IL-25 reporter mouse and found that IL-25 reporter staining here in red is very rare in the intestinal epithelium and very selectively shows up as co-staining with DCLK1 positive cells and not any other cell as part of the secretory lineage. And DCLK1 is known as this very specific marker of tough cells. These are very understudied cells for most of their lifetime that they've been known by humans. So they were actually first identified in the 1950s. And you can see from these micrographs why they're called this. They extend the projectiles out into the lumen that are highly enriched for sensory GPCRs. And they sample the lumen of the gut looking for sensory inputs that drives their depolarization. And these cells are very related to taste cells in the tongue. The reason why they are considered to be cousins of taste cells is that they share expression of this sensory G protein subunit called gusteucin that couples to G protein coupled receptors that sample the lumen of the gut. And so one of the best examples then is that succinate, a metabolite, will activate this GPCR succinate receptor on the surface of the tough cell. This drives depolarization of the tough cell through gusteucin that ultimately results in secretion of IL-25. And if we back out to the tissue level analysis of this, what is essentially is now argued as a summary of the literature is that protozoans, so particularly Trichomonas, secrete extracellular succinate. This is detected by the tough cell to drive IL-25 production. This signals to these local ILC2s that then start secreting IL-13 and IL-5. Those cytokines have pleiotropic targets, but one really interesting outcome is that IL-13 will signal back to the LGR5 positive epithelial stem cells to ramp up further tough cell differentiation. So this sort of creates a feed-forward loop whereby tough cell activation turns on ILCs that then further expands tough cells to enhance functionality. And so one of the things we were interested then in is, if we think about these paradigms that are known for fungal sensing, a lot of the work that's been done in sort of beautiful classic studies by Gordon Brown and many, many others is the idea that cell wall sugar detection by C-type lectins will turn on a number of cytokines that can drive type 1 or 3 inflammation. It's also clear from a number of studies, including some recent work looking at alternaria pore-forming toxins, as well as fungal proteases. Work from Ilion has done this, as well as Darren Weisner when he was in Bruce Klein's lab, and many other groups have shown that damage of the epithelium by proteases or toxins can result in alarmin production that can, depending on the context, drive either type 3 or type 2 inflammation. But our starting point here was, well, there's really nothing known about whether or not fungi have interfaces with tough cell activation. And so does this circuit apply in the context of fungal sensing? And so given this feed-forward loop that's known to exist, we started with a really simple question, which was that if we use Wallemia as a model, since this is known to drive these interorgan effects, can Wallemia colonization drive tough cell expansion or activation in mice? And so here we're colonizing animals and looking in the small intestine to start, since this is where most of the tough cell literature has been done. So you can see if we stain for DCLK1, there's these rare tough cells in the intestinal epithelium. And when we looked in the small intestine, which again is where most of the parasite literature has looked, we didn't see anything. So we were initially a little bit disappointed. But we continued and scanned along the intestinal tract. And one thing that we've noticed that comes up very reproducibly is that we actually get this surprising specific tough cell expansion upon colonization in the colon. And we do think part of that is that it seems to more preferentially colonize in that location. But this is surprising for a reason that I'll circle back to. We then also made tissue extracts from the colons to look at cytokine production. We can see when we colonize, we get local induction of type 2 cytokines that are usually ILC2 derived, like IL-5 and IL-13. If we then wanted to ask, are these actually tough cell dependent? And so it's been known from a number of these differentiation studies of intestinal epithelial development that PU2F3 is the master transcription factor for tough cell differentiation. So what we've done here is now do oral gavage with Wallemia into either wild-type littermate controls or PU2F3 knockout mice, which are tough cell deficient. And when we do that, we get not a complete in the case of IL-5, but we get a measurable reduction in IL-5 production when we get rid of tough cells. And then we actually get a more complete reduction of IL-13. So we do think that there is functional tough cell contributions to this local type 2 cytokine induction. Now, I'd sort of mentioned that this colon observation is surprising. And the reason why is that if you circle back to this model of feed-forward loop, whereby tough cells activate ILC2s, and then that ramps up more tough cell differentiation, there was a paper, a follow-up paper from Rich Loxley's lab, where they asked what happens if we just hyperactivate ILC2s by deleting this negative regulator A20. And when they did that, so you now make a hyperactivated ILC2, if you look in the controls, there's very few tough cells. If you look in those hyperactivated mice, you get tough cell hyperplasia in the small intestine. But when they looked across other tissues, you actually don't see that effect happening in the large intestine, like the colon. And follow-up studies that they've done, if you inject IL-4 or IL-13 systemically, you again get small intestinal expansion, but not colonic expansion. So there's been this question about how much this feed-forward loop even applies in the colon, and whether or not type 2 cytokines are sufficient or even necessary to do this. So we asked then, given that we're seeing this sort of rare phenomenon of colonic tough cell expansion in this context, is it STAT6 dependent or dependent on type 2 cytokines? And so IL-4 and 13 need to signal through the transcription factor STAT6 to drive this phenotype. So we asked what happens if we get rid of STAT6 in the system. And when we do this, comparing STAT6 knockout mice, we don't see any loss of this tough cell expansion. So if we compare back to, again, wild-type littermate controls, they see this, you get this colonic tough cell hyperplasia, and we don't see any difference when we delete STAT6. So this is acting via a very different circuit than what's been described in the small intestine. So what we're seeing then is that when we colonize with this fungus, we're getting colonic tough cell hyperplasia that's STAT6 independent, so it's a non-canonical expansion circuit. And so obviously the question is, well, what's driving this? And the hypothesis that we had was, rather than using an epithelial immune expansion circuit, could Wallemia be secreting something that's directly influencing stem cell differentiation? So to get at this, we've moved into organoid culture systems where we can grow crypts, and so now look at the effect of exogenous molecules in the absence of immune cells or neurons. And using that system, we can use positive controls, so add in type 2 cytokines, which are known to give you tough cell expansion in an organoid culture, and then take fungal supernatants and ask what we see. So what we've done here now is make an organoid culture from the mouse with just control. And you can see if we stain for DCLK1, there's almost no tough cells in just a normal organoid. If we do a positive control and treat these with IL-4, you get tough cell expansion in the organoid culture. If we then just use diluted yeast culture media as a control, that again doesn't do anything. But if we take conditioned supernatants from grown fungus, those are actually sufficient to drive this tough cell differentiation in the organoid culture. And since we'd seen in vivo that this effect was STAT6 independent, we asked whether or not the same thing was happening in this system. So we've grown STAT6 knockout organoid cultures. If we again give PBS, we see nothing. Now if we give IL-4, which should require STAT6, we don't see tough cell expansion. And then if we give non-conditioned media, we see a few tough cells, but these tough cell hyperplasia is maintained in a STAT6 knockout when we give conditioned supernatants from the fungus. So what we're seeing then is sort of, again, a mimic of the in vivo scenario, but what we found is that there might be a bioactive molecule that's directly inducing tough cell differentiation, which leads to the obvious question of, well, what is the nature of this secreted activity? And so, you can go through the list of different macromolecules, but we started off asking, is it maybe proteinaceous? And I'll say we got a little bit lucky here. We'd initially seen that the activity was heat sensitive. And then also we found that if we treat these supernatants with proteinase K coupled agarose speeds, that reduces a lot of this tough cell induction capacity. If we look at silver stains off the supernatants of these grown cultures, you can see that there's a number of proteins that are released into the media. Depending on who you ask, this is either a lot or a little. We were at least just happy to see that it wasn't a smear, which gave us some confidence that we could maybe try to purify it. And so we started off then with an old school technique, which is using ammonium sulfate precipitation cuts to basically ask where we can crash out activity and resolubilize it. And so I'm not showing the bioassays here, but we've done 40 to 50%, 50 to 60, and 60 to 70% cuts. We don't see any activity in 40 to 50, but we maintain it in the higher concentrations. So we used those that cut range as an initial purification scheme. And so what we were hoping to do then is use a biochemical approach to see if we could identify the protein coming from these supernatants. So the approach in general is to take 40 to 50% ammonium sulfate precipitates, resolubilize those pellets, dialyze, and then run on size exclusion columns, take the elution fractions off the column, put them onto organoids, measure tough cell induction capacity, and if we can get enough specific activity, try to identify the protein by mass spec. And so what I'm showing you now are just the elution fraction analyses off the column. We didn't do every single one because Hong Yu, the postdoc, would have killed me, but we at least did sequential 5 fractions off the column. And so what you can see here then is this. I'm not showing you every fraction off the column, but if you look here, 36 through 40 have no tough cell induction capacity. The next set of fractions, now activity starts coming on. That's maintained or even more so in 47 through 52. And then in the next set, that activity drops away. I mean, there's actually the same amount of protein across all these fractions. So we're at least getting some specific localizations off the column where we're seeing selective tough cell induction capacity. So as a starting point, then, we basically have taken the schematic where we've found that the activity is proteinaceous. We've done sort of an initial, semi-crude system with ammonium sulfate cuts followed by sizing. And then we basically just took these 2 active pools versus 2 inactive pools for mass spec. And then additionally, we ran silver stains off of these fractions from the column. And again, we got a little lucky here, but what we find is that in the two active pools, so 41 through 46 and 47, 52, there was this one band that came screaming high in the two active fractions. So we additionally cut that out and did mass spec on that band. And when we cross-compared the pooled mass spec to this band and then also filtered based off of proteins that have a signal peptide and in a certain size range that we knew from some cutoff filter experiments, we basically got this one protein that's called I4Y5L4, which is a 39 kilodalton protein with a signal peptide. So from there, we wanted to see if this protein is sufficient to induce tough cells in organoid cultures. So we cloned the cDNA, put it into a mammalian expression vector with an IL-2 signal peptide and a His tag, expressed this, and then enriched for using nickel NTA agarose. And if we then take the recombinant protein and put onto organoid cultures, we find that that is sufficient to induce tough cell induction in the organoid. So we do think this is the likely candidate from there. So obviously, what everyone's going to ask is, well, what's the predicted function of I4Y5L4? And this wasn't that difficult. It actually comes up right away on UniProt when you look at the protein, and it encodes a predicted secreted asparaginase, which was surprising to us. But asparaginases have been well studied, particularly E. coli-derived versions. And what they're known to do is catalyze the conversion of L-asparagine into aspartic acid and ammonia. This has been well studied because of its use as a chemotherapeutic for acute lymphocytic leukemia. And so people have done mutagenesis across the E. coli-derived version, which is used therapeutically, and it has this well-described active site that requires these two threonines here. If you mutate those to alanines, you kill catalytic activity, and you can see that motif is highly conserved in this Willemia-derived protein. So we wanted to then ask, is this a selective functionality of these fungal-derived family of proteins, or can we see a similar phenomenon with an asparaginase coming from anywhere? And so because the E. coli version is commercially available, we just purchase this and ask what we see. And so what we've done here, again, is make an organoid culture, treat with control. And then here now is treating with the E. coli-derived asparaginase. And you can see we again get this beautiful tough cell hyperplasia coming up in the organoid culture. We then wanted to ask questions about whether this is potentially due to substrate depletion or production of a product. And so what we've done now is either add back in aspartate alone or tried to rescue the asparaginase treatment with supplemental asparagine. And what we see is that when we spike back in asparagine into these cultures, that has this rescue effect on the action of asparaginase. Whereas if we put in aspartate on its own, that has no effect. So we think preliminarily that this is due to asparagine depletion. To get at that a bit more specifically, what we've done now is actually take the organoid culture media and reconstitute it from powder that doesn't have amino acids, which then lets us titrate the amount of asparagine in there. And what we found is that if we titrate the amount of asparagine in the organoid culture, that if we bring this down to lower concentrations, that is again sufficient to turn on this induction program. So what I want to just sort of back out then and talk about with these way we're thinking about this data is, I mentioned earlier in the talk that there have been these traditional modalities of fungal detection, the classic one being cell wall sugar detection through C-type lectins. This turns on a number of inflammatory cytokines that will initiate type 1 or 3 inflammation. There's also been this paradigm of protease or pore-forming toxin that essentially causes damage responses in epithelium, and this results in release of alarmins that can turn on type 2. In the case of parasites in tough cells, the argument has been that active metabolite production, so the classic example being succinate, is detected by tough cells to cause depolarization and initiate type 2 responses. What we're positing initially is that there's sort of a new modality here, which is not active metabolite production, but potentially metabolite or nutrient, and in this case amino acid depletion, might be a cue that says that there's something there that we don't want because it's competing away nutrients. And this acts as a trigger of tough cell expansion and activation that turns on the weep and sweep response. And some of the things that we're interested in understanding now are how is this actually sensed from a cell biological perspective. There's not a ton known about asparagine sensing, although some of the pathways that are out there argue for GCN2-based detection that can shut off mTORC1. We're exploring those sort of arms. There's also potential roles for amino acid transporters, or we don't want to hang our hat on it, but given a lot of the beautiful work, controversial, but from David Sabatini with specific amino acid sensors like the cestrin or castor pathways that all couple to TOR, perhaps there are unknown asparagine sensors that are coupling into these pathways. So our model then is we're thinking about nutrient depletion as a mechanism to sense fungi that sort of acts in parallel to this active metabolite sensing pathway that occurs for parasites. And some of the ongoing efforts that we have now are trying to generate asparaginase deficient fungal strains in collaboration with Rob Kramer at Dartmouth to test whether this is necessary in vivo. We're also asking whether or not some of these pathways are required for that distal airway enhancement effect. And then we're also really interested in asking questions about how specific this is to asparagine itself, and it just so happens that fungi make an asparaginase, but perhaps depletion of any given amino acid could drive a similar effect. And so these are some of the broader questions that we're trying to follow up on now in active ongoing studies. So with that, I want to wrap up and thank my lab, which has sort of been an interesting roller coaster over the last year, dealing with a transition from the NIH up to Boston. I want to make a particular shout out to Hongyu Wu, a very talented postdoc in the lab who's really single-handedly pioneered this whole project, which is a really new, interesting area for the lab. I want to thank all my colleagues back at the NIH, as well as people that I'm getting to know here at the Ragon. And with that, I'll wrap up, and I'm happy to answer questions in the chat, and then I'll also follow up with the Q&A at the end. Thanks so much.
Thank you, Dr. Dang. That's great. So it's my pleasure to introduce my colleague and second speaker of the MycoTalks session, and that's Dr. Hong Nguyen from the University of Pittsburgh. Hong, I think you can go ahead and bring up your camera. Perfect. So Dr. Nguyen is a tenured professor of medicine at the University of Pittsburgh and co-directs the Center for Healthcare Mycology and Fungal Genomics, and also serves as director of the Transplant Infectious Disease program, as well as the XCR Pathogen Laboratory at the University of Pittsburgh Medical Center. So Dr. Nguyen has a longstanding interest in both fungal and also multi-drug resistant bacterial infections, including mechanisms of antifungal drug resistance and fungal diagnostics. She's recently applied a long-read sequencing using the Oxford Nanopore technology to understand isolates that are capable, isolates of Candida that are capable of causing bloodstream infection. And so her title of her talk today is Within Host Genotypic and Phenotypic Diversity of Candida Causing Bloodstream Infections. Thanks so much, Dr. Nguyen, for giving us a seminar today.
Thank you, Dr. Cohn, for the introduction, and thank you, the committee, for inviting me to give the talk today. Can you see my slide?
Yeah, just need to go to swap
Okay.
to the, yeah.
Okay. Okay. Is it okay now? Okay, great. Good afternoon, everyone. So the long-standing belief is that most bloodstream infection and sterile infections stem from a single organism. That means that there is an expansion of the single strain that successfully passed through the bottleneck, as I shown here. This single organism hypothesis actually has shaped both the diagnostic workflow and clinical decision making for over 10 decades now. However, there have been some problem because the many microbiology lab, actually all of the common practice of the clinical microbiology lab is typically to select a single representative colony from the plate for identification and antifungal susceptibility testing. The clinician then, in turn, base their therapeutic decisions on the characteristic of this single presumed representative isolate. Over the past decade or so, there have been emerging evidence that challenges this long-standing hypothesis. In recent study, we have shown that blood culture from patients with Candida bloodstream infection can harbor distinct strain they are clonal but genotypically and phenotypically distinct. Importantly, some of these strains may differ in antifungal susceptibility and yet missed by the clinical microbiology lab. Data to date suggest that a single colony may not adequately capture the true complexity of the infection, carrying major implications for the diagnosis, treatment, and our understanding of Candida pathogenesis. So my hypothesis for following this type of the study are, number one, positive blood culture are comprised not with a single strain, but a mixed population of genotypically and phenotypically distinct microorganisms. This is particularly true and relevant for GI commensal like a Candida. Second, the antifungal treatment failure can sometimes be explained by the emergence or expansion of an already existent in the blood of the variant strain with diminished antifungal susceptibility. And the rationale behind these hypotheses are very straightforward. One is that the single colony or lineage is not representative of the entire population of the much broader microbiome community. And then the genetic and phenotypic diversity is very well recognized for over a decade at site of colonization or chronic infection, like a patient with cystic fibrosis colonizing with Pseudomonas, when the patient can be infected with several clonal but antibiotic susceptibility difference, and also for Mycobacterium tuberculosis. And lastly, once the Candida enter the bloodstream from wherever the site they come from, it is exposed to the immune defense and sometimes antifungal therapy. This pressure can drive a rapid selection and expansion of adaptive variants, resulting in a mixed population with differences in virulence, stress tolerance, and antifungal susceptibility testing. So before I present our new data, I want to briefly highlight the two recent publications from our group that laid the groundwork for the current presentation. So I start out with Candida albicans. It was recently published in the Journal of Infectious Disease, and the work is driven mainly by one of our research faculty, Dr. Hassan Badran. So the method is very simple. We obtain the blood culture from patient with candidemia. We shake it on the blood agar plate and also a chrome agar plate, and pick up to 96 colonies and save it for future whole genome sequence. So, for Candida albicans, we selected samples from 4 distinct patients, as shown here. 3 of these patients had persistent candidemia, ranging from 3 days to 10 and 13 days, respectively. One patient just have a short run of candidemia, lasted for only 1 day. For this patient with a single blood culture, we obtained 10 strains and subject them to whole genome sequence. The other 3 patients, we did 5 strains from the ignition blood culture, and then we go to the last positive blood culture and tested the 5 colonies from there. All of these underwent Illumina, and one single isolate that we obtained from the micro lab that was tested for susceptibility also underwent long read sequencing. So long story short, these the strain from individual patients are color-coded here. You can see that these strains cluster together within the patient, but are far distant from the other patient, just telling us that we did not have an outbreak going on at our hospital. When we look at the small scale genome variant, it is the finding was not very impressive. The amount of SNP and indel range anywhere between 0 to 42. None of these mutations associated with antifungal resistance. However, when we look at the large-scale genome by looking at Y-map here, you can see that we see aneuploidy, we see loss of heterozygosity, and also gene copy number variant. For the purpose of this review, I just mentioned briefly about one patient, which is patient MN. The M strain came from the ignition blood culture, and the N strain came from the blood culture isolated 13 days later on various antifungal therapy. So looking at the Y-map here, you can see that there is aneuploidy at chromosome 7. So for the first set of blood culture, 4 out of 5 had the trisomy in the chromosome 7. On the follow-up blood culture under persistent blood culture, we no longer see this kind of trisomy. So when we look at the phenotype, the trisomy lost the ability to form hyphae on the hyphoid induction plate, and it also has less biofilm formation than the euploid that mainly recover after the therapy. So we further did a phenotype by testing a competitive infection of the trisomy and the euploid strain. We use incubation in human blood ex vivo. We also test in the mouse GI colonization by testing stone, and also invasive in the GI mucosa, as well as the disseminated model of candidiasis. And what we show was that over all of these testing, the trisomy strain are more fit in this condition. And although the strain did not make much hyphae in the in vitro, in vivo, in the murine kidney model here, we did see some hyphae, but it's much less than what was seen with the euploid strain. Then we look at for the susceptibility. All of these trisomy and euploid strain are all susceptible to echinocandin with very similar echinocandin MIC. However, when we look at the tolerance, tolerance is defined as the ability for the fungus to grow on antifungal, but when we test the output, MIC of the organism output, the MIC remain the same in the susceptible range. Then we found that the euploid actually were more tolerant to echinocandin tested. We used the dot blood plate, used the refraction of a growth assay and supra MIC growth assay to evaluate the tolerance like presented by Dr. Berman's lab. So, you know, in vitro, at least the euploid strain are more tolerant to the echinocandin. Then what happened in mice? So we treated the mice with the micafungin, and then we co-treated with both the euploid and the trisomy strain. What we saw here was that without micafungin, the trisomy strain is more fit, like shown here, but with the echinocandin, then the euploid strain become more tolerant and more fit in the mice treated with micafungin. So let's go back to the patient map then. Day 0 is when the first blood culture was obtained and became positive, and then we had subsequent blood culture up to day 13. What you can see here on the original blood culture, over two-thirds of the blood culture were due by the fit trisomy 7 strain. 1 day and then 3 days after caspofungin therapy, the trisomy just almost totally disappear, down to about 7 and 2% respectively. And then now the blood culture is full of the euploid organism. And this scenario validate our in vitro study, is that euploid became more tolerant with caspofungin exposure and just persisted in blood. So in conclusion then, what we saw was that within a single blood culture of the patient, we saw different phenotype and also genotype differences. And echinocandin tolerance may account for at least some of the treatment failure in our patient. So for the second paper, it was published about a year and a half ago in Nature Communication. And we did very similar study, but we concentrate on Candida glabrata. And, in this study, we used 10 different patients, and, the finding was very similar, except for the following, that, in Candida glabrata, we did not exhibit the large-scale genomic changes like aneuploidy, like we saw in Candida albicans. Instead, what we found was that within host diversity, it was driven by small-scale variant of SNP and indel that correlated with antifungal resistance. So out of these two, of the 10 patients, 2 failed fluconazole therapy. And then on the subsequent blood culture, we obtained fluconazole resistance. Then we went back to the ignition blood culture, and we found a small a percentage of colony, fluconazole resistant colony that were already resistant there. And the mechanism of antifungal resistance or mutation within the 11 gene in one patient, and then the efflux, PDR1 and MDR1 in the other patient. So together then, these results challenge the current single organism hypothesis. If validated, then the findings suggest that both the clinical and the microlab practice may need to consider for a microbiome population, and it may change the pathogenesis of invasive candidiasis. To share with you. As you know, here, as you all know, Candida auris is an emerging multi-drug resistant pathogen. Unlike Candida albicans glabrata and other Candida species, except for Candida parapsilosis, these tend to be an exogenous rather than the GI commensal. So it primarily a skin colonizer, and this is the setting for multiple outbreaks throughout the world. And, the outbreak thus far tell us that most of these Candida auris generally clonal. However, for the past 2 years, there have been increasing in small report that showed within host diversity. So, people found mixed strain, so different clade in single patient, and one of the patient actually had SNP variant over time. And the SNP difference and indel among isolate from the same patient. And then in one patient, resistance developed to echinocandin and amphotericin B, and the resistance were found to be due to mutation in the minor subpopulation. So I'm going to show you data on 2 different cases that we saw. The first patient is a Candida auris fungemia from a patient that was transferred to our hospital from Nevada. And we all know that Nevada has had an outbreak of a clade 3 Candida auris. He came in and then developed a bloodstream infection. So we put the line, the patient did just well, responded to the echinocandin therapy and survived. Because of the clonal nature of the Candida auris, we decided just to pick 41 randomly chosen strain. The morphology is different, so we pick as many different morphology as we can, and all of these underwent along with ONP sequencing. So long story short, you can see here all of these strain from the patient are all very, very genomically related and related to the clade 3. However, in 3 out of these 41 strains, we found a possible circular chromosome that we are still working on it to prove. And also in 6 out of these 41 strains, they had chromosome 3 aneuploidy. And the aneuploidy was confirmed by 5 different genes that are located in chromosome 3. And then, for the phenotype, we saw that the diploid Candida auris had much less aggregation compared with the euploid, and also has much less adherence using the PMI, sweats medium, and also in collaboration with Janet Nett in Wisconsin, we also did the model in skin adherence, and the same pattern was true in all 3 models. The case 2 is more complicated and very interesting to me. This is a case of Candida auris, a chest wound infection after lung transplant. So at the transplant, we routinely did the BAL. So, it can target the antibacterial and antifungal prophylaxis. And we found, by surprise, that the BAL was colonized with Candida auris. 2 days later, the patient developed infected chest wound hematoma. That it turned out to be a big deal because the patient subsequently developed a persistent infection that involved the entire mediastinum and caused a mediastinitis. So we have sequenced the strain that were colonized in the BAL and the 2 disease-associated strain. But for today's talk, I just going to present to you the data that we have from the BAL itself. So this work was done by Xiaoxi Chang from our lab. What Xiaoxi did was he did the whole genome sequence. This time we did Illumina, and then we did a long read sequencing for one of the index strain. And as you can see here, all of these strains were clonal, very, very closely related genomically, and it's also related to the clade 1 strain that we obtained from the CDC. So, they are very clonal. Then, using wipe map on the entire 20 strains, we found only 1 that are totally different than the other one. And the finding is when we first look at it, it looked like a disease aneuploidy. But the headache stopped because when we look at the number of copy variant here, actually these what's so-called aneuploidy actually is not, because the copy number is non-integer. It's less than 2. So it's between 1.3 to 1.5. When we saw this, the first question came to our mind is that, are we dealing with the mixed population of Candida auris in a single strain, which we call A1 here? So, before we do everything further, we check for growth rate. And, the A1, again, A1 is, we think that there is a mixture of a lot of different strain in there. And A2, which seemed to us that it's only 1 strain, the growth rate is the same. However, when we carry the lifespan up to about 40 days, then, the S1, the one with the aneuploid like on web map, actually had less growth benefit than the S2. So to look at the possibility of mixed colony, then we spread the A1, which has the copy number variant compared with A2. And, we waited for 4 days and, so it's different to tell, but you can see from here, there are larger colonies along with the smaller colonies. And the larger colony here are different and larger than what we see here in A2. And A2 look like it's a homogeneous lawn of similar colonies. Then we did again, but this time we stopped the incubation at 2 days. And it became very clear. There are small and large colony here. So, we try to isolate the large colony and the small colony to have a single colony so we can do further phenotype. However, we spread it for five different passage, we continue to see a mixed phenotype. So from the large colony, we see the majority of large colony, but you can see here, there are smaller colony. And in the small colony, we have a large colony as well. And despite the 5 passages, so we get like a large to large, and we get large to continue to spread, we continue to have the mixed colony. So the question that we ask is, and then, when we look at these, so the question we ask, is it switching phenomenon that we are seeing? So what we see here is in the small, so this is, we pass it twice. You can see here, compared with the large colony, the small colony has a very irregular border compared with the large colony that it's mainly smooth border. So to look for the switching, we put this isolate on the fluoxin B plate, and you can see the large, the A2 is mainly white, and then the A1 is pink. Then, we put more colonies on that, and then now it's clear that we are dealing truly with the switching from the colonies that we obtain in vivo. So in the large, big is, larger colony, it's, and in our hand at day 7, on the colony look pink. So, these are smooth colony, but on the A1, from the, after 2 passage, you see small, you see large, pink, and we also see white pink. And on top of that, we have the sector morphology that we see both pink and white. Then, from the small colony, we look under the microscope, and the small colony actually give us the larger cell size than the large colony on the plate. It gives us a smaller cell size, and this is the A2 for frequencies. And in both of these microscope slide, you can see a mixture of large and small as well. So, since large colony has been linked to ploidy differences, the next question we ask, are we dealing with ploidy difference? So, we get these isolate that we obtain from day 8, day 30, and day 50, and we subject it to Illumina sequencing. As you can see here, whatever the copy number variant here that we saw in S1, it disappears. But whatever on the right side of the chromosome, it persists. And it seems like from day 8 forward, we have a stable organism that, the genotype hasn't changed. So we look for diploidy. So we use 2 strains, one from the CDC, given to us by the CDC, and the other one by our colleague, Robert, I forgot his last name, I'm so sorry. But as you can hear, for the diploid, then you see 2 peaks. One is at the 1C peak and the 2C peak, and it's about 9K at the location of about 19K here. And for the tetraploid, the flow showed the shift to the right, and it's about 20K, so it's the bone of the 10K, and 40K, which is the bone of the 20K. So what happened to our isolate? Looking at A2, which is look on the chromosome, look the same. So as you can see here, there is a peak at 5, a peak at about 8. So this is half of what the diploid that I show you from here. So this is a haploid type of strain. Then we look at the T8. This is the stable strain that we got after multiple, after the 8-day passage. You can see that the 2 peak at around 10 and 20. So this for sure is the diploid strain. And the A1 is somewhere between the haploid and the diploid. And you can see a small peak here of the C1, and then the larger peak later. So we think that the A1 based on the flow is a true mix of the organism. And then we run some susceptibility to micafungin. The A2 is more resistant than the A1, and whether it's large or small, it's more susceptible than the A2. And these ploidy differences show that it has less biofilm information than for the regular haploid A2. So in conclusion, Candida auris, just like albicans and glabrata, exhibit genetic and phenotypic diversity within the clinical culture. In patient 1, we saw chromosome 3 aneuploidy, either fragmented, segmented, or the whole chromosome 3. Patient 2, we saw a mixed cell population of at least 2 karyotypes that are distinct and it associated with phenotypic switching and differences in morphology, lifespan, and biofilm formation. So altogether then, positive culture may be comprised mixed population of genetically and phenotypically Candida species. Type of the predominant genetic diversity, at least in our study, may differ between Candida species. In Candida albicans, we saw mainly aneuploidies, LOH, and large-scale genome variant. Candida glabrata, we saw mainly SNP and indel, and Candida auris, we saw switch phenotype and ploidy differences. Thank you very much for your attention, and I would like to thank my lab who have done all the work that I present today. So thank you again for your attention.
Thank you. Thank you for the fantastic presentation. And now it's the most fun time here, Q&A. So the way we're gonna do that, I will start with questions to Eric Dank, and I'll be combining questions that are in the same direction. So maybe sometimes we'll have 2 questions answered in one shot. And please, for all the participants, please add your questions to the Q&A. So I think we'll alternate with Jay after that. So the first question to Eric. So many people actually are complimenting you both for the talk and for your new position, but here you can see that in the chat who is saying what, but we will go specifically about the questions now. So couple of questions are very similar. So people are very interested because you saw actually aspergillus in E. coli. So the questions are, how conserved that is across fungal species is one of the questions. The other one is more, if you look at species similar to Wallemia, what is the situation there? So how conserved is the phenomenon?
Yeah. So asparaginases are expressed by almost all fungi. The question is more whether or not they have one in the genome that has a signal peptide or not. So in general molds, so most Aspergillus species, Penicillium, Fusarium, et cetera, Cladosporium, they all have signal peptide containing asparaginases. So they tend to secrete quite a bit of activity. And this has been scanned a lot because of this use as a cancer drug. People have been looking for different sources of asparaginases for a while. What I can say is we've done some preliminary experiments taking soups from non-Wallemia species, like certain Aspergillus strains, and we can see the same phenomenon happening there. Now, in terms of global patterns about which ones are allergenic versus not, I don't want to overstate that per se, because there are other mechanisms to trigger things, but there is this general pattern that molds, which we do know tend to be very type 2 triggering, do have very conserved expression of these asparaginases that are secreted.
Yeah. Okay. Very nice. Jay, do you want to go or I?
Sure.
Yeah.
Yes, we have a question in the Q&A for Hong. So the question revolves around since these are clinical isolates, what do you think the role of prior antifungal exposure is, or actually immunological pressure? Although I think a lot of these patients are probably transplant recipients. So what do you think is regulating this aneuploid state in vivo?
So for the aneuploidy in in Candida albicans. So our lab, we started with Candida glabrata frogs, and we saw all of these resistant popping out in the ignition culture. So for Candida albicans and Candida auris, we specifically picked the sample from the patient without antecedent antifungal for the past 3 years. So, you know, the aneuploidy that we saw in Candida albicans and in Candida auris from people that have not been treated with antifungals.
Great, thank you. Hiten.
Yeah, so I'll go with the next question, which is actually linked to what, Eric, to what you actually started discussing here. So there is a suggestion that the aspergillus circuit being co-opted as a way to accommodate the fungus by a way of real estate expansion instead of pathogenic clearance. Have you thought about that? And do you see overall enlargement of the colon in your
Yeah,
model?
so the enlargements are, so I'm trying to think if we've actually measured it. We haven't noticed that overtly, right? In terms of co-option, you know, it's always the debate about, you know, is it microbe-driven or host-driven? I mean, I'm biased here, but I think because of the conservation across most species, even ones that aren't necessarily mammalian adapted, I think this is more of a more ancient pathway for fungi. Probably these asperginases seem to be very upregulated by nitrogen starvation, and so it's a nitrogen scavenging mechanism. That doesn't mean that it doesn't play any role in adapting to host conditions potentially, but I don't think it's a circuit that's evolved to colonize hosts. But I'm perfectly willing to be proven wrong on that.
All right, so interesting things to do. Jay?
Yeah, Hung, how is aneuploidy regulating echinocandin susceptibility mechanistically, do you think?
I think it depends on which chromosome and which gene it affects. So, you know, for Candida albicans, we know a lot about the genome because it has been curated, so it helps us. For Candida auris, it's tough because, you know, it virgin. But I think aneuploidy does not imply that this is a tolerance or resistance. It depends on which chromosome and which gene is associated with the aneuploidy.
Yeah. So I don't know for glucan synthetase 1, what chromosome is that on 7 in Canada, or do you think maybe there's other mechanisms of resistance that or susceptibility where you know access to the GSC1 enzyme maybe, you know, impeded by some other issues in the cell wall, right, that allow interaction?
So for Candida auris, the majority of the clade are resistant to fluconazole, right? So, and the resistance is mainly due to R11. In Candida auris, at least on clade 3, it belongs in the chromosome 3. What we saw was that, so now, you know, we are looking for the gene that associated with the aneuploidy and the adherence, because that's what we are after, because for the pathogenesis. And, you know, what we found was in Candida auris, it's ALS4, which is on that chromosome, and we did RNA-seq. The data I haven't shown because
Yeah, I was going to ask you about transcriptome.
Yeah. So it's upregulated. So, you know, again, it tells us that it depends on which chromosome and which gene that are involved.
And do you see? Have you had the opportunity to look at environmental isolates, or do you think there's something about the bloodstream isolates specifically that that is allowing a?
I wish I can.
Yeah.
At our center, you know, the micro lab screen using a PCR. So that, you know, it's the work, and they're afraid of spreading, and the work it's just so tedious. So I don't have the skin isolate, but I got a skin from people in Chicago, and you know most of them are clonal. So there is something that, you know, once the Candida auris get into the human host, It's
Yeah.
not to change.
Interesting. Thank you.
You're welcome.
Right, so question for Eric. I will combine two again here. So the first question is if actually the tough cells in the colon versus the small intestine are different in terms of sensing. And in the small intestine organoids treated with aspergillase, do tough cells expand? And the second question is, why only tough cells, do other cells also, cell lineages also respond to this mechanism?
Yeah, those are great. Those are great questions. So there are differences in the colonic tough cells that have been described. So there's this ATOH1 dependent expansion circuit that we still need to test, actually. I think, why the expansion is colon specific. So, one argument would be people have looked at relative asparagine levels across the intestinal tract, and they are lower in the colon compared to the small intestine, except in germ-free, where they're actually higher and just steady everywhere. So it is possible that because of that lower concentration, it's more sensitive to these enzyme activities. The other argument might just be that it colonizes there better, and we do see that. We've done organoids from both small intestine and colon, and we see the exact same thing. So we don't think it's something specific about the stem cell programming that allows that to happen. What's still curious is, I spent all this mental exercise on why IL-4 circuit doesn't apply to the colon in vivo, but it actually does work in organoid cultures. So, and I've talked to, Jakob von Moltke and Rich Loxley about this, and it's not clear why, because they've seen the same thing. So there, there's something in vivo where that's repressed, but then it can still be observed in vitro, and I think that's not understood, but that's not our main goal.
So it might be not the cells, but the environment indirectly or directly is doing something. But it's not the cells themselves.
Right. And then in terms of, do we see other lineages? Yes, that's a great question, because there is this general secretory lineage that kicks on, and you do see all that coming on when you give IL-4 or IL-13. So we've looked at that. So if we do Willemia treatment or asparaginase treatment, we've done PCR looking at lineage factors for Paneth, goblet, enteroendocrine. So far and away, the most induced gene signature is tough cells. You get very little Paneth and goblet cell induction, but you do get a little bit of enteroendocrine induction. So it's not 100% tough cell specific, but that's definitely the most specifically upregulated.
But the tough cells might be the sensors, basically.
Yeah.
If you look at all that.
Right.
Okay, thanks.
Yeah, so there's a follow-up, Hong, about, so you think that the aneuploidy has to be specific to the chromosome or even in this context of copy number variation, potentially locus specific to confirm resistance?
So it's
or
not
changes.
like so so aneuploidy is duplication of the chromosome, right?
Right.
So it can duplicate the gene, so the gene effect is amplified. But in that chromosome, they may have transcriptional factors that act on other chromosomes as well. So I think that I have to rephrase it to make it very clear that the aneuploidy might or might not lead to resistance. It depends on what gene and what transcriptional factors are there on that chromosome.
And then what about azole and echinocandin resistance in the colony variants? Is how divergent is it between the small colony variants and the large colony?
So in the about Candida auris.
Yes.
Okay, Candida auris, all of the strain was susceptible to echinocandin, but when you look at the MIC, the small colony tend to be even more susceptible. So the MIC is like a 0.06. The other one is by e-test, it's go down to about 0.006 or 0.008. So it's even more susceptible. We did not see any resistance there, at least not yet. We are still screening.
Okay. Thank you.
Okay, so there is another question for Eric, which goes, in the same spot, I think. So somebody's wondering if you can actually measure aspergine in different parts of the gastrointestinal tract. So since all the tough cells respond similarly, could it be basically the substrate?
Yeah, and that's what I was alluding to in the last question. So it's been, there was this, actually you were on that paper, Ilya. There was a group from Cornell that did that study where they were doing mass spec along the intestinal tract for different amino acid concentrations, and you can see.
Yeah, but I have to read the question, so we have to discuss here.
But what was seen in that paper is certainly that the asparagine levels are higher in the proximal small intestine, and then they drop as you go down to the colon. They're still present there, but it's at lower concentrations in SPF mice. And then if you put them in germ-free conditions, then everything shoots back up. So there are these local modulations that are dependent on the environment there. So, again, like back to what I was saying before, the specificity could have to do with that concentration difference. It also could have to do with just the burden difference of where the bug is. And, we haven't totally uncoupled that.
Yeah. Okay. Jay, do you go or I go with the second question?
Yeah, well, either way, go ahead, Elaine, while you...
Okay, so I can go with the second question here. It's still back to this, evolutionary idea of what fungi might be expressing it. So can you say that dimorphic fungi have the same mechanism of action?
Dimorphics, yeah. We haven't looked. And so we haven't done those experiments with like Coxy or Histo or anything like that. We've done it with Candida and some other things, and we don't see that at least in yeast form. We haven't tried it in pseudohyphal form because I'm worried the Candida lysin. We might have to do it in a ECU1 knockout so the Candida lysin doesn't just kill the cultures. So I can't answer that yet. But it's possible. We just, I think we're viewing this more as a model to start getting into that biology. But the question is, as you start to extend across looking at different species, where do the patterns start to emerge? And I think that's going to be where it's going to get fun.
Yeah. Thanks.
Yeah, so we have a question for Dr. Sese from the NIH Clinical Center, who's done a lot of work on pneumocystis genomics. And his question is, do you know if the aneuploidy observed in the Candida auris isolates are centromere mediated?
I don't know.
Not sure.
No,
Yeah.
sorry. Yeah.
So as a director of, somebody who's done so much clinical infectious disease as well as genomics over the last few years, what do you think the emerging technologies in a clinical mycology lab for determining resistance is going to be now that we have all these tools like long read sequencing and also just traditional antimicrobial susceptibility? Do you see a role for AI or artificial intelligence in the clinical mycology lab?
So now, with the nanopore, once you can sequence, you get the reason back within a day. So, it much less work than doing the antifungal susceptibility testing. For echinocandin, it might be feasible because of the FKS. It's very specific. Although now, more and more data emerge that there might be other secondary mechanism of resistance that we don't know yet. For fluconazole, I think it will be very difficult because there are so many genes, and some we even have to do expression data to look for the resistance. So I think if it works for the echinocandin for now. But it's going to be very difficult to just to use the genomics for the ozone, and for bacteria as well, because I run that lab. Also, it just there's so much resistance here. I have to do it, and we do beta-lactamase, NDM plus ABCD, NDM, OXA, and those does not cover for chromosomal resistance, right? Like borin. So the PCR or the BiHealth, but I don't think it can ever replace the susceptibility testing.
Yeah, great, thanks, I actually have a question for Dr. Dang, actually, about so I think Bart Lambrecht's lab showed several years ago that that IL-25 is also post-translationally activated by proteases. So do you think there's fungal proteases that could not only cause tough cell differentiation, but also potentially make IL-25 more potent through post-translational enzymatic modifications?
It certainly, was it 25 or was it 33 that he was arguing in that context?
But I think 25 too.
Yeah. So
I may be misquoting it. Maybe David Corey's work, but yeah, I think
no, it's one of those. Yeah, that's right. Yeah, no, that's certainly possible, and there could be sort of two-step models here, right? Where, there's the initial production, but then the proteases amplify, and I mean, Ilyan's recent Kazakhstania work too, where kind of shows that these Kazakhstani Assyrian proteases can cleave 33 to provide more activation. So that sort of might be a conserved pattern that's emerging in that area, right?
It's very hard to prove though in vivo.
Yeah.
The cost uses.
And speaking of Bard, I mean, he also has some data out there, that there's the question of cleaving to more activate some of these alarmins, but then there's the other question of, is there any specificity to inactivating more type 1 polarizing cytokines?
Yeah.
And I actually think there's some data kind of emerging on that topic too in that realm.
Yeah, I mean, at least in the murine model, I mean, pneumocystis actually drives a strong type 2 response in the lung, and then Sergio Vargas has shown that in humans where he's looked at lungs of infants that were, unfortunate demise due to something else like a motor vehicle accident. In the pneumocystis fungal burden is really correlated with goblet cell hyperplasia in those human lungs. So,
right.
But I'm not aware of the mechanism of how pneumocystis regulates a type 2 response.
Right.
Relative contributions of IL-33 versus 25.
Right. I mean, in general, tough cells, they're not very common in the lower airways.
Right.
They are there in the trachea, right? But again, that's mostly been looked at in the context of these type 2 cytokine expansion circuits, which don't seem to apply in the lung. But, we haven't looked at all there in that organ yet. And, would you think about things like Aspergillus species that definitely do secrete asparaginases, is any of that playing a role in non-type 2 cytokine dependent expansion? Would be kind of interesting to look at.
Yeah, that would be very cool. And you're collaborating with the right person to make those mutants of Aspergillus. So that will be fun. So with that, we're at the end of our MycoTalks series for November. Thanks for the speakers. That was very stimulating talks and discussion after that. And always fun to co-chair with Jay. And thank you all. And we'll see you in December with other chairs and other speakers.
Thanks, everybody.
Okay, thank you. Bye-bye.
Bye.